information is strictly transferred or decoded firstly from DNA to RNA in the process called transcription, then from RNA to protein by specific decoding according to the principle of genetic code in the process called translation. And so, in fact, it's a very interesting question which molecule of these three, for example, could evolve as the first one in the evolution. And still, most of people tend to think that it could be RNA, and this led to the hypothesis of the RNA world. Though, of course, nobody can really, and perhaps nobody will be able maybe to show this experimentally, because this RNA world or any other world just vanished. But anyway, so according to RNA world hypothesis, so DNA was produced from RNA by maybe reverse transcription or maybe indirectly or somehow. And so the whole variety of modern proteins were produced in the process of translation, perhaps. And of course, first translation must have been mediated by RNA itself. And we can see that both in current, in modern life, translation is very important because it produces the whole variety of proteins which perform now the vast majority of the vital functions. And as well as translation was very important in the evolution because it did create the, most probably, the whole variety of the same proteins. Well, what is translation? Very roughly speaking, it consists of four main steps. Initiation, elongation, that is the growth of polypeptide chain. Termination, that is the ending of the protein synthesis. And the so-called ribosomal recycling, which involves the transfer of the ribosomes from the termination point from the synthesis again to the initiation start point to start a new cycle of translation. So it's kind of a cycle. And just to demonstrate the complexity of the modern translation, I will briefly show the most complicated and the most subjected to regulation step of translation, that is the translational initiation in eukaryotic cell. So basically eukaryotic mRNA looks like this. It has the cap structure at the 5' and it has some 5' non-translated region, non-coding region. It has AUG start codon, then it has some coding region, and then somewhere it has stop codon, not depicted here, then 3' non-coding region, and poly A tail. So this is a typical eukaryotic mRNA. And then we have a lot of different protein initiation factors. Even kind of mentioning all of them will take perhaps the time of this presentation. So some cap binding factors, some RNA helicase, and so on, ternary complex. So it's just to show the complexity of modern system. And the interaction between, for example, initiation factor 3 and 4F promotes the loading of the 40S small ribosomal subunit onto the mRNA. And this is 43S complex. So then we have poly A binding protein, which makes this kind of a closed loop by interacting with poly A tail and with some of these initiation factors. Well, and after this small ribosomal loading onto mRNA, the following steps of initiation are the scanning, when this initiation complex scans the 5' non-coding region until it reaches and recognizes the first AUG start codon, from which the elongation starts after the large ribosomal subunit joins this complex. Then the termination occurs when a ribosome encounters the stop codon. And then it's supposed that ribosomes should be somehow recycled, or in fact physically transferred again to the 5' end to start the new round of translation. So this is a very complicated system. I have not depicted here even the whole set of the initiation protein factors. Otherwise it would require, I think, one more presentation. But apparently, early translation systems must have been much more simple, because it's obviously that proteins did not exist at some point on this planet, at least, for example, in the RNA world. But at the same time, some modern RNA molecules, for example, RNA molecules of viruses, have some features which can be considered as molecular relics or fossils of the RNA world, if it existed. Which characteristics should we search or should we look for when we are looking at the modern RNA molecules, and which of these features should be are considered as satisfying to the criteria of the RNA world? Of course, it's protein independence. If RNA is not dependent on protein, then it can serve as kind of model, or at least example, of what could be the descendants, or at least resemblance of the RNA world. Quite often, and in fact, quite often RNA molecules are not just a stretch of nucleotides. So they form quite complex secondary structures, kind of stem loops. And for example, such secondary structures are absolutely required for any RNA catalytic activity known so far. And RNA secondary structures also can facilitate protecting RNA from the degradation. Well, the picture of translation initiation which I showed, so it was a kind of typical eukaryotic-like translation initiation. But many RNA viruses many modern viruses use completely non-canonical translational mechanisms, and so they have some structural elements which are also non-canonical. And some of these elements are, for example, internal ribosomal entry sites, so-called irises. And these irises present RNA secondary structures, something like this. They can be present either in the 5' encoding region or in the intergenic region if the virus has several open reading frames, several genes. And these structures are recognized directly by the translation initiation complex, because RNA of such viruses lacks the cap structure. And translation in such cases is 5' and independent. So that's why it's called internal initiation. And I will give just some illustrations of these internal ribosomal entry sites, which could perhaps evolve at different points in evolution. For example, irises of Picorna virida viruses. So they look like this. So it's a kind of set of stem loops, secondary structures, and AUG start codon somewhere here. And here in this picture just shows the protein requirement, how these irises depend on the initiation factors. And in fact, these irises require essentially the same set of the protein initiation factors for their translation as any cellular cap-dependent RNA requires. For example, a very important scaffolding factor 4G, RNA helicase 4A, and just 43S complex. So, except for 3 factors, which are not needed here. Another example of another degree of protein dependence is presented by Flavivirida, including the hepatitis C virus, for example. So this initiation mechanism requires only 2 protein factors. So this is a secondary structure of this internal ribosomal entry site and the open reading frame with the AUG start codon. But viruses from the family Decistroviridae have 2 genes, so 2 open reading frames in the genome. And they have even 2 internal ribosomal entry sites. One is located in the 5' non-coding region, and the second one is located in the intergenic region. And the second iris, translational initiation from the second iris, doesn't require any protein initiation factors at all, as well as it does not need initiatory methionine tRNA, because the translation here starts from the alanine codon, preceded by the CCU codon depicted here. And this is a secondary structure of this intergenic region iris of one of these viruses, that is cricket paralysis virus. So the colored nucleotides show just conserved nucleotides among the about 10 species of this family. And so we see quite intensive secondary structure and quite a lot of base pairings, so between some stem loops, this kind of a tertiary structure. And but the length of this whole iris element is just 180 nucleotides. So it's even shorter than the shortest viroid. And for example, so here is the first decoded codon, alanine codon. And for example, there is a base pairing in the region of the codon immediately preceding the first decoded codon. And in some experiments, mutations were made to destroy this base pairing in the region of that codon. And this completely abolished the activity of this internal ribosomal entry site. Why is it so? This is how usual cellular RNA is positioned inside the ribosome. So the AUG start codon is always in the P site of the ribosome. the next second codon is in the A site. So the initiatory methionine tRNA, aminoacylated with methionine, goes into the P site. This is directed by the base pairing interaction between the AUG codon and the anticodon stem loop of the tRNA. In the same, and of course everything is mediated by proteins here. In the same way, the second tRNA goes into the A site, and it carries the corresponding amino acid also. Then the peptide bond forms. The next step here is the so-called peptide transfer, when the first tRNA is getting deacylated. So this bond here is broken then. And after that, the anticodon, the free anticodon, acceptor stem of the tRNA interacts with the E site of the ribosome, and this is considered to promote the next step, that is so-called translocation, when the ribosome slides down to the slides along the mRNA molecule in a way that the first codon turns out to be out of the reaction center of the ribosome, and the second codon goes into the P site. And this interaction between the acceptor stem, free acceptor stem of tRNA, and E site is important for translocation. But of course it's mediated by proteins. What do we have in the case of cricket pearlitis virus? So as I have already said, so even such a relatively short RNA element, less than 200 base pairs, can function in a protein-independent way, in a completely protein-independent way. And that element of secondary structure, which was destroyed by mutation, it mimics the anticodon stem of the tRNA, and it base pairs with the first codon located in the P site. In fact, this codon is not decoded, so it's kind of not codon. And so this interaction provides tRNA mimic-like interaction provides the correct positioning of the viral RNA inside the ribosome. And this step is, as I have already said, completely protein independent. And this internal ribosomal entry site also is considered to contain another important secondary structure element, which probably interacts with the E site of the ribosome. And this interaction promotes the translocation. But translocation in this case does not require the preformed peptide bond. Well, obviously, because there is no tRNA here with no amino acid. But this is the illustration. But translocation here still requires some proteins, at least one protein. But still, at least at the step of initiation, this process is completely protein independent. And so this is a very good illustration of how this internal ribosomal entry site of virus, of this cricket pearlitis virus, may be considered as the relic of the RNA world. And then some experiments with RNase P digestion were performed, and they showed that all the three groups, that internal ribosomal entry sites from all the three groups of viruses which I mentioned, they contain tRNA-like structure, because RNase-P digests RNA exclusively in the region of the tRNA-like structure. At least structurally it should be tRNA-like structure. Not necessarily it should have the same sequence. And this takes us to the importance of tRNA structure in the evolution of the protein synthesis. So genomic tag hypothesis was proposed by Alan Weiner and Hansen Metals. So this postulated that perhaps all RNA molecules could be perhaps all the RNA molecules in the RNA world could be tagged with the tRNA structures. It would be needed to tag those molecules which should be replicated. And the examples, and this idea is supported by the observation that some modern plant viruses and bacterial viruses do have such tRNA-like structure at the 3' end. But then probably some RNA ribozyme could evolve to cleave this, to detach this tRNA structure from the RNA, to distinguish between those RNA molecules which should be replicated and between those which serve as RNA enzymes. And then, if such free tRNA-like molecules could evolve a capacity to base pair with the future mRNA molecule, and if they happen to be together in close vicinity on this RNA, and if they gain the capacity to be amino-acylated, then the peptide bond could form with help of RNA enzyme or spontaneously. It could be a protoribosome. And then also we could speculate that this kind of detached free tRNA-like structure could also could be transferred to the 5' end of the mRNA molecule, and it could become covalently linked to the 5' end, and that's why this could be the explanation why, the speculation why the tRNA-like structure is found in the viruses, modern viruses. And just some illustrations of viruses for which tRNA-like structure is very important for translation. So this is turnip yellow mosaic virus. It encodes some proteins. It has 5' cap structure. It has 3' tRNA-like structure. This structure looks like this. It's valine accepting tRNA-like structure, so it's capable to be amino-acylated by valine. But it was demonstrated experimentally that this tRNA-like structure, when it's amino-acylated, can donate its valine residue as the first amino acid of one of the viral proteins, which is the polyprotein, very long protein. And this mechanism is completely 5' end and cap independent, although this RNA contains cap. And this is in fact, I think, quite a very good illustration of how this translational mechanism of current modern virus can resemble, or at this can be similar to the idea of the proto-ribosome, because the tRNA-like structure donates its amino acid to be incorporated into the protein. So it plays the role in translation. Another virus, bromosaic virus, which has a genome composed of 3 RNA molecules and also 1 subgenomic RNA. It also, each of these molecules is kept at the 5' end. Each of them has tRNA-like structure. In this case, it's tyrosine accepting tRNA-like structure. So it looks like this. And it was demonstrated that this tRNA-like structure is also very important for translation of these viral proteins. Although it was not shown that this tRNA-like structure can donate its tyrosine residue to any of the viral proteins. Perhaps it stimulates translation by a different mechanism. It's not known yet. Some viruses have also 5', 3' and interaction. But unlike the eukaryotic mRNAs, this interaction is not presents just a direct base pairing between the RNA fragments. And for quite many viruses, it was experimentally shown that the interaction between such kind of secondary structures, one of them is located in the 5' non-coding region, and the other one located in the 3' non-coding region, and these are hydrogen bonds, base pairing between stretches of nucleotides in these structures. So this interaction is critically required for translation, perhaps at the step of translation initiation, when this kind of closed loop-like structure is formed, which perhaps effectively transfer ribosomes to a new cycle of translation. So I was talking, I mentioned internal ribosomal entry sites having quite well developed secondary structures. But some viruses have such kind of internal ribosomal entry sites which don't have very well developed secondary structure, but instead they function as the translational regulatory elements, as internal ribosomal entry sites, just as the stretches of nucleotides, that is, on the level of primary structure. And if it has ever been shown that these translational mechanisms mediated such a simple structure or simple viruses are protein independent, then we could also consider these mechanisms as models or at least resemblances of the vanished RNA world. And instead of conclusion, I would just say that modern viruses present quite a wide variety of translational mechanisms which can serve as some kind of hints or illustrations of what could be in the RNA world in sense of the evolution of the protein synthesis. And if somebody is interested in original articles, which I have mentioned here, and I have a list here. Okay. Thank you.
the absence of a world which generates proteins, is that actually an obvious thing? I mean, once you have RNA which has the capability of generating proteins, and you have the right building blocks in place, what does it take to prevent the formation of proteins? That is, to escape the pure RNA world. Is it the incapability of having DNA?
As a first molecule.
No, okay, if you have the RNA world as a molecule at one point in the RNA world, wouldn't that immediately be ready to synthesize proteins?
I think if we assume that the genetic code could not evolve kind of immediately, so that it's, I think, even statistically would be the most probable event, so that most of the at least first RNA molecules in the RNA world, if it existed, did not code any proteins. They were just non-coding. They were just some kind of replicating stretches of nucleotides.
See, yeah.
Yeah, so what has been experimentally shown is that RNA molecule can, as a ribosome, catalyze acylation reaction, so the peptide synthesis, so that the product is RNA peptide conjugate, not separate peptide molecule, but so it can elongate its own chain via the peptide chain. So that has been experimented. So if that is what you ask, that is it possible?
And the peptide chain would be a protein then?
Sorry?
The peptide chain.
Peptide chain. Peptide is a small part of protein, yes.
Yes. So in that sense, it would produce proteins on its own at the end of its.
To the 5' end of RNA chain. So it has been by chemical selection. So it has been created this kind of ribosomes which are possible to which are able to elongate, add a few amino acids.
Yeah.
Before the RNA world was invented, or at least become the most favorable early phase, there was always two groups of people. One said protein first, the other said DNA or nucleic acids first. And I think those people who favored protein first showed already that number one, amino acids can be easily made abiotically. Number two, you can polymerize these amino acids abiotically to some chains which have also some catalytic functions. Why can't we think that in the RNA world, which is more or less still a mixture of organic molecules, there is also the abiotic production of proteins, which by chance have a certain function which is helping the RNA world to catalyze and move, so that there are some interactions between these abiotically formed, what they call protein lines, and the RNA molecules. Can we think something like that?
Well, probably not. But at least 3 years ago, one Japanese group has shown that experimentally that at least one step of what they called reverse translation is possible.
Mm-hmm.
I don't remember now.
Like proteins?
I mean, like kind of from proteins to RNA. It was not the complete, it was not the complete way from proteins to RNA, it was just one step. I don't remember now chemically what was this, but at least, but No, it was just this one small, very small step. And I have not seen any more papers showing like this.
This is in conclusion to Gregor's suggestion. So I think it is quite commonly thought that the early reacting ecosystem was a mixture of different types of molecules, including randomly polymerized peptides. And of course, like if they come by random polymerization of amino acids, then there is no accuracy of polymerization. So it is quite chance and random interactions. But probably because the peptide chains would have been easy to make, they would have been available for some kind of study.
So this is not so closely related, but people always speak about catalytic RNA. That's true that RNA really is catalytic and very many catalysts have been created. But we also should remember that DNA is catalytic. So nowadays in laboratories also for DNA enzymes have been created, which are relatively short, only a 15 nucleotide loop of DNA is able to catalyze the cleavage of phosphorylase carbon. So we have to remember that what is catalysis? It's a spectrum. It's dependent on time. So proteins are certainly efficient catalysts. RNA less efficient catalyst, but it is a catalyst. DNA, when it is single stranded, may be a less efficient catalyst than RNA, but it is a catalyst. So it depends how much time we have to do different things.
Are you in favor of DNA PC?
No.
Yes.
I am in favor of parallel development of RNA and DNA in a way so that they have different functions. One to store the genetic information and the other one to do something else. And I think that RNA has played a major role to create proteins through RNA peptide conjugates. That I believe. But I don't quite believe in such a way that we have had quite developed RNA work without any other form of storage of information in addition to RNA. Because the ability of RNA to store information, to my opinion, is so limited that very few things could have been developed only on that basis.
But what about modern viruses? They sometimes have genomes reaching up to 12,000 nucleotides.
Yes. But still it is not very much if you have to encode a lot of proteins.
Oh, yeah. We compare with human genome, for example, yeah, of course.
May I use this? The question was proposed, what is catalysis? I have studied the typology of chemical reaction in the context of this project and noticed that only two types of catalysis are known in chemistry. And it seems to me that the theory of catalysis is undeveloped, very much undeveloped. The chemists have not paid attention to the logical possibilities and the types of reactions. They have only predicted all the time new and different reactions, but not systematized them. I have done such a systematic typology of catalysis on the basis of algebraic and logical possibilities of reaction mechanisms and reaction structures. And I have 12 different types of catalysis. And they are not all, because when we come to more complex systems of reactions, like in molecular